It's great to be here today. This is my first time attending this conference, and I think this is just a really great group and have just been so impressed by the talks that I've heard today. I'm not going to be talking about mechanical ants. Instead, I'm going to be trying to take you through the idea that cells inside your body are essentially acting as machines. And one of the neuroscientists that I take some inspiration from is Warren McCulloch, and as well as his graduate student Walter Pitts. And I love this picture from one of these early papers. This is in the 1950s. And what they did is they used these northern leopard frogs to try and understand neurocircuits and how they are able to compute logic. On the left here, we have, this is a retina inside of the frog, and you can see the rods and cones up here at the top, and you can see all these other cells. And if you look at these cells long enough, or short enough, you'll see that they all look a little bit different from each other, right? They all have a very different structure. They are all shaped a lot different from each other. And they even have these different processes, these axons and these dendrites of different lengths and even diameters. And their structure is conferring some of their function. And this is something that they observed. And he wanted, like the saying that he will say over and over if you watch his videos or videos of him talking, he'll talk about how he wanted to understand how man would know a number. Right? So numbers are very logical. So he wanted to understand how these circuits inside your brain can essentially confer that logic. And so he brought on Walter Pitts. Walter Pitts was a mathematician, and the two of them studied these neurocircuitry by looking at the anatomy of the frog eye and also looking at the electrophysiology of all the cells in this pathway, and tried to deduce what the logic was that was occurring in order for a frog's eye to know a number. Well, not really, but I guess a frog's eye to interpret sight. And this is something that I would say we're still trying to figure out, right? And that's been pointed out by other neuroscientists here today. Okay, so how do these cells get their structure in the first place? This amorphous cell on the left, or round cell, I guess, it's got these different cytoskeletal elements in it. These are the three basic ones that we talk about in cellular biology. We have our actin microfilaments on top, our intermediate filaments, and our microtubules. You'll notice that the actin microfilaments, that those are out in the cortex of the cell. They're really on the outer edge of the cell, and they are what are conferring the actual shape of the cell. And they're highly dynamic. They're moving all the time. On the right here, I'm showing a specific cell type. This is a microglia. Microglia are the immune cells of the brain. And they change their morphology all the time. And yours are actually changing right now in your brain as you're listening. On the top left, this is a highly ramified microglia that has these finger-like structures. And this is what they're like at rest in a non-inflamed condition. And then if you get ill, or if you have a concussion, or there's some sort of insult to your body, there's these transmitters that are released in your periphery and your circulatory system called cytokines, and they can get into the brain, and they can actually cause these microglia to move the actin around inside of their cells and cause them to change from that ramified structure in the top left down to that amoeboid structure in the bottom right. And so that's one of the things that we study in our laboratory. We study that transition and how these cells are behaving. So I said actin is the major player. This is the ribbon structure for actin. It's a globular protein. And you'll notice right there in the middle, that's adenosine triphosphate. So this is an energy molecule. In this case, actin is using ATP as a molecular switch. When it binds to ATP, it makes it so it has a higher affinity for other globules of actin. Okay, so we have our fancy animation. Okay, so you'll get a bunch of globules together, and they'll start to form into filamentous actin. And this filamentous actin looks a little bit more like this. It's a winding structure. And as that ATP gets consumed, all of a sudden the affinity of globular actin for its neighbors, it will actually decrease. And so it'll fall off. So you'll notice on the left-hand side, we have some actin falling off. And on the right-hand side, we have more actin being added on. In this way, these actin filaments become highly dynamic. And so if you were to look inside your intestines, you all just had lunch, a delicious lunch. Thank you for the lunch, by the way. You have these cells inside your gut, these epithelial cells, and on the apical surface or top surface of these cells, you have all these microvilli that look like little fingers. And the length of those microvilli is directly proportional to the amount of actin, filamentous actin, that's filling up inside of those microvilli. And you can see that on the right there. As the actin extends, the microvilli get longer. Right? And so this is an example of your cells being dynamic and moving inside of your gut so that they can increase the surface area of absorption. On the bottom right here, I have a cross-section of a cell. You can think of this as being one of those microvilli just kind of sticking out, but you could also think of it as any cell that's going to be moving. And so here we have this cell, and it's got this filament of actin, and just above it, we have these myosin heads. These are motor proteins that will bind onto actin and cause it to move. And these also use ATP. In this case, they're using it more as a sort of battery. And while it's being pulled back, we're still getting more actin that's added onto the front of that filament. And you can actually visualize this occurring. So this cell on the left, this cell is not moving, but the actin inside of it is moving as it's being pulled away from the front in a process that we call retrograde flow. Get that to go one more time so you can kind of see it moving like little waves within the cell. So when you want the cell to finally engage, you actually have to add another component. And this component, these are called integrin receptors. Another name for these are focal adhesions. So these allow the cell to actually adhere to a substrate and stick to it, right? So I like to think of these as kind of being like little tiny treads on tires that allow the cell to like stick to a surface and pull along. So once those focal adhesions engage, now the cell can move along. Okay, so I'm trying to draw some analogies here to start to make it seem more like these are like machines. Right, these tiny little cells. So on the top left, I have some actin and myosin components. I'm going to liken that to a motor. And then I have the focal adhesions on the bottom left, and I'd liken those to a tire. I'm going to add a third component on here, receptors. And receptors are proteins that are going to be detecting specific ligands, right? They're looking for some sort of molecular pattern. And these ones I liken to a sort of transmitter. It allows the cell to know exactly where it's actually going to be going towards, right? So that it could have some sort of goal-directed movement. On the top right here, I have a cell. And in this case, this cell is going to be an immune cell. Immune cells move like crazy. They want to move towards an area that's been damaged so they can clean it up. And you'll notice that all surrounding that cell, it's got receptors everywhere. Right? You need to have these receptors everywhere in the cell so the cell knows where to go. So it can detect a higher concentration of ligand in a specific spot and move towards it. In the lower part, I have the actin and myosin complexes. Of course, those are building and those become a sort of cytoskeletal anchor for the cell to move along with. That's that same actin-myosin complex is also able to contract and pull the back end of the cell forward. In fact, those same sort of actin-myosin complexes are what allow your muscles to contract when you move your arms, right, or your legs. And then you have your focal adhesions on the bottom right of that front cell, and those focal adhesions are forming at the front of the cell, and at the same time the cell removes those focal adhesions at the back so it can propel itself forward. So here we have these tiny little machines moving throughout your body. And you can see these in nature. So if after today you decide to do this, you could go fishing in the Provo River. Maybe they have fish, I don't know. And while you're catching the fish, if it gets a little injury, then pull out your microscope and start scraping away some of the cells around the scales. And some of the cells that you'll scrape away are these cells called keratocytes. These are wound healing cells. And these keratocytes, when you put it under your microscope, it'll look a little bit like this. Let's see if that goes. Okay, so they'll be moving around underneath your microscope, and they're looking for some specific guidance cue. They're looking for where the wound is, right, on the fish, and they would be moving towards it. And these keratocytes are terribly interesting. They have all the components for moving, and they have some specific direction that they're wanting to move, or goal they're wanting to move towards. And you'll notice at the back on the left of it, that's the nucleus. If you were to just completely cut the nucleus out, all the components, all the mechanical components are still there, and that will still keep moving, right? As a small machine, it doesn't even need the nucleus to move, right? It has all the protein components to move. Now you have something similar in your brain. I already mentioned these before. These are microglia. This is not your brain. This is a rodent brain. Anyone bring their pet rodents? Probably not. Okay, so that's a rodent brain, and the red box is over an area called the striatum. This is an area that's involved in conditioned learning and habit formation. This is where dopamine is released when you hear people talk about dopamine. And if you were to zoom in on this area, you would see that it's just absolutely filled with microglia, these immune cells that are constantly moving inside of your brain. If you were to zoom in on these immune cells, this is what they'd look like. And we'll do a little 3D rotation. Ooh, okay, so they're three-dimensional, right? It's not just a flat image like you're used to seeing with microglia. They're constantly moving, so I'll repeat that video here so you can see that again. Over a 30-minute period, you'll see it move just constantly. And we see these move all the time. In our preparation, we have rodents that have green fluorescent protein inside of these cells, and you can see them dancing around. And they're not just dancing aimlessly. Each of those processes has receptors on it that are listening for specific guidance cues. And if you were to overlay all the movement across a 30-minute period, that's what we're getting on this far right-hand side. Right, so there's a lot of movement that's happening within these cells just while you're going about your day-to-day. Think about that in terms of plasticity. You've heard that term before, right? This is about as plastic as it gets. Now, of course, these are important for wound healing. So here you'll see on the left-hand side, this is a fluorescent label right here with the microglia. In the middle section here, this is a non-fluorescent label, so you're seeing structural data. And you'll actually see a dead neuron. You have to repeat this, but there's a dead neuron right here. You see that movement right there? And that dead neuron, this is in the overlay right here, on the dead neuron, you'll see it get pulled towards the microglia as the microglia is phagocytosing or eating up that dead neuron. So it's kind of cool watching this happen. So that's called phagocytosis, which means to eat. Another type of behavior that they exhibit all the time is called trogocytosis, which means to nibble. Right? And so they're constantly nibbling away at little synapses throughout the day when it's like, oh, you don't need to know their phone number anymore, and it just kind of gets rid of it, right? And maybe you forgot where you placed your hammer, and so it nibbles up that synapse. Okay, so that's your phagocytic activity. And again, just to emphasize that this is very much a receptor-mediated activity. So here's an experiment where we take a little glass electrode, that's what's shown here on the top right, and we filled up that glass electrode with some ATP. And ATP, in addition to being an energy molecule and molecular switch, it's also a damage pattern. So when cells die, they explode and ATP goes everywhere. And so your immune cells, basically all of them, are just really into ATP, and they'll just start going towards it. So that's what you're going to see in the fluorescent channel. Just imagine the glass electrode where it says local ATP application. Right, so those processes from those microglia are just moving in, and they're trying to stop the damage because they don't want other cells to get damaged. Okay, so that's a not very specific guidance cue because like all microglia have ATP receptors. But if you were to look at another neurotransmitter, So dopamine is a very common neurotransmitter. It's the one that we study in our laboratory. And this is in the striatum where there's dopamine release. So let's see if these microglia respond to dopamine. And the long and boring answer is the short and boring answer is they don't. Okay, so you're not seeing a lot of responses here. And then as my students kept doing these experiments, they found that a very small population of microglia would move. And then you'll actually see this one climb up inside of the electrode, which is kind of crazy. So some of the cells are really responsive. And in some cases, we think that microglia can be reprogrammed, and in some cases be programmed to sense after specific neurotransmitters, right? And maybe in some cases that is pathological, right? Like not a good thing. So we're still trying to figure out what this means. And this is very latent breaking. Don't tell any of my competitors. Okay, so this sort of cellular motility that we see, it's not specific to microglia. It's actually like something that we see in pretty much every type of cell. So of course, neurons would be included. So here are two neurons. So on the left is an axon, and it's communicating with a neuron postsynaptically. And the fluorescent signal that you're seeing here is actin that's fluorescing. And so you just saw a bright spot there. That was as a synapse was forming between these two neurons in a dish. This is not in a brain. This is in a cultured preparation. And this actin formation that's occurring here, it's very much dependent on the electrical activity inside of the cell. So on the right there, I have a diagram showing some synapses. On the top, we have the presynaptic terminal. You can think of that as the neuron on the left. On the bottom, we have the postsynaptic dendrite, which would be the cell on the right. And as the cell on the left releases a neurotransmitter such as glutamate, it's again behaving very much like a machine. It's got all these vesicles filled with neurotransmitter amongst other things. And those things get released out into the extrasynaptic space. And then the cell on the postsynaptic side, it sees that as a guidance signal and will move towards it. And a lot of times when us neuroscientists are getting in a room and we're talking about this stuff, everyone's talking about glutamate, but you would expect to see this sort of behavior for other transmitters as well, right? And I would also say that you would expect to see it in other tissue as well, not just brain. This is a common function. We just kind of get fixated on the brain because it's so fun. Just to fixate on the brain one more time for my last slide. So this is a technique called Brainbow, where you can put fluorescent protein in lots of different cells, and it's very pretty to look at, but there is a purpose, so you can see networks as a whole and how they're connected, and you can look at individual cells and actually see how one is different from the other. This is in the olfactory bulb of a mouse, so this is involved in smell for this specific circuit. And the thing that I want to focus on here is that, yes, our brains are incredibly complex. We have all these different neuron types, right? These are all shaped a little bit different from each other. Those are different types of neurons. And they're going to be connected to each other and networked in very specialized ways. And so when McCulloch was looking at the circuits in the frog retina and trying to understand the logic there, that was a sensible thing to do. Right? But the thing that you'll also notice is that there's a lot of missing spots in this tissue. There's just as much not there as there is there. And that's going to be made up of all these other cells that aren't necessarily neurons. In some cases, they're going to be part of the circulatory system, the blood flow. In some cases, they're going to be the glue, is what we were calling it for so long, right? The glia, the microglia, the astrocytes. And we're discovering more and more that these are just as important for maintaining the balance of these networks. And then the last thing that I'll end on is that It's really a whole body. When we're thinking about how our experience shapes our, I don't know, what our experience actually is. Earlier today we did those breathing exercises. That was part of our experience. And when we're talking about drugs of abuse in my lab, we think about things outside of the brain, right? When we're talking about alcohol, that's what we study. We think of the liver. We think of the damage that it's causing there and the immune cells that are being activated and the cytokines that are being released. And we talk about all the different things that are happening in the periphery that can then contribute to your experience, including the deleterious effects of these drugs. Thank you for your time. Our research is funded by NIAAA, and we're required to mention that, of course. These are the students that performed those microscopy experiments in the top left. And yeah, thank you for your time.